Okay, hopefully saya akan share nanti. Alright.
Okay, terima kasih doktor.
Okay. So Bismillahirrahmanirrahim. So I think again, can you see my slide? Is it okay?
Yes doktor,
Yes,
visible.
betul.
Saya nampak
Okay.
doktor.
So, alright. So Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. So I think we'll start on another topic. So basically, my topic is not uh, solely on the arrangement of the what we call the course outline. So basically, this topic liposome will be, I think, in the course outline with the end of the last topic within the course outline. But I think because each individual topic is independent to each other, so I think it's okay for me to start with liposome. Okay, so for the topic, we will learn about liposomes as drug delivery vehicles. Okay, so some introduction. So liposome or vesicles are cell-like spherical aggregates formed by self-assembling of amphiphilic molecules such as phospholipid. Okay, amphiphilics, as I mentioned previously in our class, amphiphilic is molecule that contain both hydrophobic and hydrophilic properties. Okay, it both have lipophilic, which is hydrophobic, and hydrophilics with that towards solubilization towards water. Okay, in terms of its properties such as phospholipid. Okay, phospholipid, I think I show you here. This is example of phospholipid. Okay, phospholipid is amphiphilic molecules, okay, that contain phosphate in the head of it compound or molecules, which is hydrophilic. Okay, and then they have it have two fatty acids within the change, that is hydrophobic. Okay, non-polar and the polar here. So for phospholipid, it will form a linear, okay, a linear type of some kind of vesicle. So later on we will see what this phospholipid can forms. Okay, so it first discovered by Bengham in 1960s, and it is an ideal drug delivery system because of their high degree of biocompatibility and their ability to encapsulate a large amount of material inside the vesicles. The lamella vesicles, which is in the round shapes, are important as model for biomembranes for drug formulation, for gene delivery, and vehicle for delivering various agents used in cancer therapy. So liposomes are especially effective in delivering drugs to phagocytes of the immune system. Okay, this is the example of my cells, some kind of my cells. Okay, actually it is in the form of sphere. Okay, this is the cutoff of the sphere. As you can see, it depends on what type of drugs, okay, that we want to encapsulate, okay, within the system. So if you see here in the middle, okay, the hydrophilic head, okay, will be together, will be bound together, so it will be in the outer surface, okay, outer area of the whole system. Meanwhile, the hydrophobic part will be in the inner segment of the system. So based on, I think you are aware about polar will attract to polar, non-polar will attract to non-polar. So it is like uh, dissolve dissolve light. Okay, some kind of like okay. So in here, the hydrophobic tail will improve or will encapsulate more hydrophobic segment or hydrophobic drugs within the system. So in here, due to that interaction, so the numbers of encapsulation of drug will increase. So it will difficult, in contrast, it will be difficult if we only use hydrophilic segment or hydrophilic system within that particular compound, that it can hardly encapsulate hydrophobic drug because the difficulties or the theoretically or the compound, the configuration of compound itself that not suitable to be high encapsulation compared to this kind of drug delivery system. Okay, so liposome drugs delivery system will improve the therapeutic index and viability, increase efficiency, and reduce toxicity. So reduce toxicity and increase efficiency, meaning that it will increase the delivery due to the numbers of encapsulation drugs that got quite high that can be delivered at the end in the minimum of therapeutic efficiencies. Meanwhile, reduce toxicity. In the end, the person, that particular person, okay, that will be delivered, okay, that we delivered any kind of drugs, that we will have a specific range of concentration. The delivery will not exceed the maximum concentration, so that will reduce toxicity and increase efficiency. That's the idea or advantages of liposomes. Okay, they have been used as carrier for anti-cancer drugs, antimicrobial agents, the delivery of micromolecule DNA and proteins, diabetes, and cardiovascular diseases. It's not only delivering drugs, some kind of drugs of anti-inflammation, anti-cancer, but it can deliver DNA and proteins. Okay, many liposomal formulations have been in clinical trials and a few successful liposome-based drugs has been approved by FDA. So basically, the study have been extensively done by researchers, okay, on how this kind of liposome to be used in drug delivery application. And in terms of research, this kind of uh, molecules, this kind of drug cargo agent have been widely studied and be applied in numbers of drugs. However, in terms of commercialization, it's still low. Okay, it's still low, but the potential is there and the record and the data is there in terms of research. But the commercialization is still low because mainly due to the stability and the production of large number of liposome that quite difficult to be handled. That's the major, we can say that the major drawbacks in commercialization of this kind of drug delivery system compared to others. Okay. However, the therapeutic potential of liposome drug delivery system has not been fully reached due to design application problem with drug delivery system. Okay, this is the drawbacks why the commercialization is quite difficult for this kind of drug delivery system. First, because of the stability, time of release, cost of preparation, short shelf life, and poor interaction with certain drugs. Okay, meaning that not all drugs type of drug can be delivered or can be encapsulated or can subsequently cut in or successfully delivered, okay, using this kind of drug delivery system. Maybe it can be highly encapsulated, but it can't be released towards the specific site, so at the end it will no use lah, right? So something like that. So it is still difficult to selectively increase the viability of the drugs at the target tissue while maintaining stability during circulation. So next we will look at a conventional liposome. Okay, conventional meaning that without the actual modification. So later on we will look at a modification of conventional liposome that we want to increase the its effectiveness, okay, of this conventional liposome CL. Okay, conventional liposomes are lipid vesicles composed of various phospholipid, uh, glycolipids, and other lipids without derivatization to increase the circulation time. Okay, without increase the circulation time. So this is some example the compound that we use. We have the SPC PC1 here. Okay, the compound one, and then this is PE, distereopoietyl ethanolamine number 2. Okay, this again, this is the compound as which part is hydrophobic, which part is the hydrophilic. So you can relate about this with this actual compound. Okay, right. So among the most important phospholipid, okay, PC, which is the molecule number 1, usually form a bilayer structure here, bilayer structure. Okay, meanwhile PE tends to form micelles. Micelles, example the sphere one, that previously I show you, okay, or inverted hexagonal structure because of its small head group. Because of its strong propensity to form bilayer structure of vesicle, PC is the most extensively studied phospholipid, especially as drug delivery carrier. Okay, this one. Okay, right. Okay, PE has been studied extensively because protonation of the amino group can lead to transition to a lamella packing mode. And PE is the only natural amino acid that can be modified easily in the head group region. Okay, that's the idea, or we can say that these two example is most common conventional liposome that been not uh, been altered, okay, without derivatization. Okay, meaning conventional liposome is the lipid used without derivatization. Okay, right. However, the uses of natural membrane lipids have been limited by difficulties in their fabrication and their poor stability. So fabrication is related towards the how to, its difficulties in how to and on us for us to handle the reaction, the yield and so on. About the poor stability is it can be degrade by our body. So that's related towards the poor stability in the whole system, and also related towards the uh, poor stability in reaction with the drugs. Okay, because of liposome contain both hydrophobic and hydrophilic layers. Alternatively, hydrophobic molecules can be contained within the bilayer and water-soluble material within the aqueous compartment. Highly nonpolar drug can be trapped within the nonpolar bilayer, whereas more polar molecules can be encapsulated within aqueous cavity. Right, inside liposome, a drug is protected from the cellular enzyme that would destroy it. Okay, example like this, the drug will be here. Imagine that the drug will be in the inner layer. Okay, or example of this sphere. So the drug will be in the inner layer. So the outside sphere will be protection for the drugs. So that's the idea here. Okay, inside the liposome, okay, the drug will be protected from the cellular enzyme that would destroy it. Okay, this increase the lifetime of the drug in the body and therefore reduce the dosage required obtain at a desired effect, meaning the minimum therapeutic effect, and this will increase the efficiency and also reduce toxicity. That's the reason. Conventional liposome have been proven to be poor carrier for drug because they are readily broken by contact with other surfaces. So that's the problem about the conventional liposome, and easily degraded by cellular enzymes, meaning it rated toward its stability. The problem with stability here. They can also be easily disrupted by pH, the changes in pH, temperature, or salt strength. So other properties or environment changes within the whole system can degrade the conventional liposome. And again, it's related about the stability and suffer from low stability and rapid clearance from blood. So in the end, the drug can't be delivered towards the target before the targeted site. So the drug will be released before it arrives to the targeted site. So that's the problem about the conventional liposome. And the major limitation of using conventional liposomes are drug delivery vehicles are first rapid clearance from blood because of the degradation, restricted control or encapsulated molecule release. So because due to the low stability, it can be released before it arrives towards the targeted area. So that's another problem. So and then low and non-reproducible drug loading because of being degraded, so it's non-reproducible. Physical and chemical instability, large-scale sterile preparation. So it's about the last one here is about the preparation of the liposome itself. Okay, right. So next we will look at the stabilized liposome. So stabilized liposome is, we can say, an enhanced liposome from conventional liposome towards our targeted application. So we will see how we can alter, okay, make a modification towards the conventional liposome to ensure to increase. We cannot say it already been stabilized, so at the end it cannot be degraded, or it cannot be it is the perfect drug delivery. No, but it compared to conventional liposome, it is more stable, okay, to deliver specific drugs towards our targeted application. Okay, so because of stability problems with conventional liposome, scientists have sought many methods to stabilize them. So these are three main methods that have been used to stabilize conventional liposome. First is the statically stabilized liposome, which are sometimes called stealth liposome. So later on, we will look why these um, modification of liposome, we call it stealth liposome, which is synthetic polymers are used for steric stabilization. Second one, another approach involves cross-linking membrane component covalently or by the polymerization of polymerizable lipids. And the third one is utilize unusually stable acbacterial membrane lipid mimics. Okay, so first, let's just look at strictly stabilized liposome. Okay, protecting the liposome surface by polyethylene glycol or PEG or other polar surface ligands such as carbohydrate has led to development of stealth liposome. Okay, here. So this is the phospholipid, and then here is the polyethylene glycol. Okay, here is the polyethylene glycol. So the we all know that polymer is about the molecule or molecule weight. Okay, so it's not all the molecule weight can be attached towards the PEG or of the PEG towards the phospholipid. So the range of molecule weight is within 1000 to 5000 only, because this 1000 to 5000 PEG molecule weight, it been approved first because it's been approved by FDA. Number two, that this range is suitable to be inserted to human body and within specific application. So at the end, the selection of molecule weight of PEG to be attached to phospholipid will depends on the type of application you want to apply. Okay. Right. These long circulation liposome have improved pharmacokinetic compared to conventional liposome. Surface component on stealth liposome prevent the liposome from sticking to each other as well as to blood cells or to vascular wall. They are invisible to immune system and have shown promising result in cancer therapy. Okay, they are invisible to immune system, so they will increase the stability of that particular liposome, and this invisible to immune system give a name to the stealth liposome. Okay, they are less prone to uptake by liver. They can therefore remain in circulation longer than conventional liposome. Right. The resulting liposome show decreased uptake into the mononuclear pathologic system, increased circulation half-life, increased stability to the content leakage, dose-independent pharmacokinetic, right? Statically stabilized liposome can stay in the blood 200 times longer than conventional liposome. So we can tackle the first right to rapid clearance from blood. So in compared to conventional liposome, it is better. Okay. Increase circulation life, increase ability content. Okay. Next, incorporation of PEG and other hydrophilic polymers into lipid barriers give rise to sterically stabilized liposome that exhibit reduced blood clearance and concomitant changes in tissue distribution. Right. Site-specific drug delivery can be achieved by using strictly stabilized drug carrier where ligands bearing targeting information are attached on the carrier surface or when and phase transition induced by external stimulus. We will see this type of strictly stabilized liposome later on. We can say it is triggered type of liposome, meaning it can be delivered to a specific sites. Okay, when it's arrives at specific site, the stimulus, okay, some kind of outside external stimulus, some kind of difference in heat, different in pH, it will open up the liposomes, and that particular drug will be delivered towards that targeted area. We will see later on. Okay, studies with PEG liposomes in patients have shown that liposome can induce side effects such as flushing and tightness of the chest. And PEG liposome were cleared relatively rapidly from the bloodstream without the exposed long circulation property. Right. Next, second one is polymerization stabilized vesicles. Okay, fatty acid with polymerizable group have been used to prepare polymerizable vesicles. Okay, here remember we will look later on. Phospholipid containing the acetylene in fatty acid chain and the acylatic lipid with polar head group have been synthesized and characterized. Other polymerizable groups that have been explored include butadiene, terminal vinyls, and methochlorine functionalities and acrylates. Okay, here's the difference. Okay, here, here, here is the you see the difference is here, butadienes. Okay, this is okay. Okay, the example 4 is the difference in butadiene, and then methocryl here, and then diacetylene. Okay, diacetylene here. Okay, alkynes, the differences, that's the differences. Okay, the head is almost the same. Okay, phospholipid and two fatty acid. Okay, the differences is within the change of fatty acid. Okay, right. One general drawbacks to use of this phospholipid in application is instability again, right? So the conventional in terms of conventional liposome, so this vesicle tend to lase easily and leak material that are trapped. This related with the conventional liposome. So the cross-linking of monomer unit after formation of vesicle will lead to stabilized vesicle with improved stability. Okay, right. The third one is bacterial membrane. Bacteria and some gram-positive bacteria can survive in extreme environments such as high temperature, organic solvent, and extreme pH. Okay, this kind of external that's can be degrade or can degrade can that the main problem that lowering the stability of conventional liposomes. Okay, these organisms survive by synthesizing membrane lipids containing very long fatty acid chains that go through the entire membrane instead of just from the middle of the membrane to the outside. Okay, next we look at sorry, before that, you have any question? You understand until now? Any question?
Yes, Doctor. So far,
Yep.
yes, Doctor.
Yes, meaning you have question or you understand?
Understand.
Oh, okay, okay, okay. Right.
Understand,
So,
Doctor.
can I continue? We can tanya
Uh,
yang kosong.
Boleh, Doctor. Tafadhal
All right.
mashkur,
So,
Doctor.
Boleh, Doctor.
okay. So we look at cationic liposomes. New approach to the treatment of disease where aberrant genes are replaced with functional ones. Basically, once inside liposome, the polar DNA molecule can traverse the protective membrane of the target cells. It is also protected from nucleases, enzyme that degrade DNA. The suitable carrier should be able to encapsulate the gene protected from nucleases, deliver it to the cells, release it, and then be degraded safely. So this is how the idea in delivering DNA proteins. Okay, so by using the kinetic liposome, it is suitable to as a drug, as a DNA carrier for targeted or specific cells. Okay, the most popular synthetic liposome carriers are often cationic lipid-based system, which having amino groups in the head group region or quaternary ammonium salts. Okay, under physiological pH, the amino groups are protonated and therefore have positive charges. Okay, the problem associated with cationic liposome delivery system is that they are rapidly cleared from the blood before they can deliver the genes to the targeted site. So again, it's also related with the stability. Also, toxicity and low transfection efficiency are major barriers limiting their clinical application. And this is some example of cationic liposome. Sorry, dogma was commercialized as lipofacin as one-to-one mixture with dope, some kind of drugs. Okay, and has been widely used to transfect a variety of animals and plant eukaryotic cells and lipopolyamine dogs. Okay, okay, here has also been commercialized as transfectam and has been shown to transfect many animal cells efficiently. Okay, So meaning here, cationic liposome we use as drug as DNA delivery. Okay, so because of the readily or the combination of DNA protein within the cationic liposome is quite good. So they use this kind of liposome to deliver this kind of DNA or proteins. Okay. However, because of low stability, the cationic liposome still in more or less not widely commercially available. Okay. So next is liposome with triggered release functionalities. Okay, as mentioned before, this liposome like some kind of drug delivery system can be uh, triggered. Okay, from the outside or stimulus. Okay, meaning the changes in that specific site or outside stimulus, some kind of usually the temperature. Okay, and then we can see the differences in ultrasound, meaning that we have the static a specific site, and then we produce ultrasound, that meaning that I previously mentioned that ultrasound can increase the heat of your skins. So we know that there is different changes or changes in terms of your skin temperature. So it will increase that particular liposome can be degrade. Okay, can be open up. Okay, you know the sphere can be open up, or the bilayer can be split up, okay, by the changes of this heat. So when this kind of stimulus appear or occur, or this open up or being triggered, so the drug will be released towards that particular site. So that's the idea. It's almost the same in differences, okay, in terms of pH. Okay, we know that cancer have different type of pH, and tumor has more acidic than our normal pH, skin pH. So when it arrive to that particular cancer part or tumor, so that this particular liposome will be triggered that particular changes in terms of acidity. So it will be triggered in terms of differences in changes in pH. So it will be open up. Okay, so that drugs will be released toward the targeted site. So that's the idea, basic idea on how liposome will trigger release functionalities. Okay, first we look at the thermosensitive liposome related towards the changes in temperature. Temperature sensitive liposome leak more readily above the phase transition temperature of their membrane lipid. These liposomes are designed to be stable up to 37 degrees Celsius, but with the breakdown as they pass to an area of the body where the temperature is above 40 degrees Celsius. Okay, such sites include the interior of tumor or an area subjected to external local heating. Again, it also can be related because some kind of tumor have specific or quite higher temperature compared to normal temperature, and to external local heating, maybe due to usually we use ultrasound because ultrasound can will not kill the our skins. Okay, so that's the main idea of using uh, ultrasound to increase the temperature. Okay, thermosensitive liposome have found application in the treatment of many prosthetic hyperplasia. This is example of uh, commercialized use of thermosensitive liposome, right? So animal studies have shown that heat-sensitive liposome can improve chemotherapy delivery to tumors. Thermosensitive liposome represent one of the advanced delivery methods, right? Or for anti-cancer agents. However, they require external heat to activate the liposome surrounding, including the tumor area. Okay, this is not applicable to the treatment distant metastasis. It is technically difficult to produce thermosensitive liposome that can retain encapsulated drugs. Okay, again, at the end, the problem is on how we want to produce this kind of uh, liposome. Okay, because technically it can be done, but in terms of bigger yield, higher yield, in terms of the ability of instrumentation, okay, the ability of the whole system And then is one we once we want to make it commercially available, so we also must think about the shelf life, okay, the transportation. Either it easily can be transferred one place to another, okay. That's also difficulties that acquire in terms of development of liposomes as the delivery cargo, okay. Next, we look at the pH sensitive liposomes. The endosomal compartment has an acidic, okay, within 5.5, and elevated acidity is also observed in tumor cells. And pH sensitive liposome were initially designed to exploit the acidic environment of tumor to trigger the stabilization of liposome membrane. However, tumor insultum is often not acidic enough, so that's another problem. The most extensively studied pH sensitive liposome are various PE, okay. PSPE has derivative, okay. PE is conventional liposome, but PE derivative is stabilized, more stabilized liposome in combination with mildly acidic amphiphilus. The PE head group has been primary amino group which can be protonated in low pH condition. Okay, the rationale is that after protonation of PE at lower pH, we mean acidic area, the membrane packing will be disrupted and the content will be released. Okay, the idea is almost the same like thermosensitive liposome. Okay, a few systems utilizing acid labile or cage liposome have been developed. All right. Next, these are mainly based on derivative of naturally available lipids such as modified PE and plasmalogens. Synthetic to ester derivative have also been developed as acid cleavable lipids. Okay, this is another example of reducible. Okay, the idea of pH sensitive liposome. The anhydride modified dope such as aniline and citrazonyl dope are sensitive to pH 5.5 to 6.5. The liposome hydrolyzed rapidly to give dope. Okay. One drawbacks of this natural lipid-based system is that they are difficult to synthesize. Again, it's difficult to synthesize and purify in large quantities. So that's why the idea of using liposome and drug delivery system is in commercialization is still limited. But if you search the research or study, okay, in terms of liposome or drug delivery system has been wide, it's not new. It's been widely studied. Different kind of liposome have been developed. Different kind of drug delivery drugs have been used to test this kind of liposome effectiveness. But in the end, for us, it's still difficult for us to see that this liposome to see it commercially available. Okay, right. PE is not available in large quantities. Okay, and it difficult to isolate or synthesize. In order for acid-sensitive system to be practical, the problem of producing material on large scale or at a reasonable cost has to be addressed. Okay, right. So this is some example of pH-sensitive liposome. Another example. Right, next one, we have light-sensitive liposome. We call it photopolymerization of lipid tails in mixed lipid system that result in paste separation, thus releasing the content. Okay, azobenzene isomerization synthesize photooxidation cleavage of the lipid tail of plasmalogen and diplasmalogen, and the result is increased membrane permeability and photocleavage of dope derived and OVC dope. This is example of light sensitive liposome. This is how it works. Okay, this is the drugs and OVC. Okay, it will attach with the drug with the liposome. Okay, and then through the UV light, it will release the drugs. Okay, so that the photosensitive liposome. Okay, that's the idea of photosensitive liposome. Next, the fourth one, we have redox triggered liposome. So redox triggered PEG conjugate containing cysteine cleavage dithiobenzyl urethane linkage. The rationale for this approach is that the cytoplasm has a lower redox potential. Okay, this cytoplasm that attach to that particular liposome, and substantially more molecule will free sulfur-hydride groups. Okay, liposome containing around 3 mole of compound compound 33 and 97% of dope were stable in plasma for over a day, but released most of their contents within an hour in the presence of diethyltritol. Okay, this is example of redox triggered liposomes. Okay, right. Again, do you have any question about triggered liposomes?
Saya ada question, doktor.
No question, Doctor.
Okay, no question. Right? Alhamdulillah.
Ada
Okay.
doktor.
Dr. Fashihah
Sorry.
ada soalan. Ada soalan.
Ya, Fasihah.
Ada soalan.
Dia type
Tak.
Apa
Doktor,
soalannya apa?
Saya nak tanya dekat slide thermosensitive tadi.
Okay. Okay.
Yang satu lagi. Tak. Okay ni. Dia cakap yang thermosensitive liposome ni require external heat. Maksudnya macam mana tu?
Okay, this one as I mentioned before. Okay, example, this is our we know that will be injected in the drug within the circulation or within our veins. Okay, if we can be delivered towards we cannot directly move it to anywhere. It will go anywhere in our body within the vein. However, if we have that specific targeted application, example, we have a tumor here, so we know we use that thermosensitive light that will be triggered or be opened up. We break down that, but if there is high temperature, okay, I mentioned before, I think, okay, meaning the above 40 degrees Celsius. Okay, once, okay, example, we put ultrasound here, so we know that the particular site that we want to make sure that this liposome release that this targeted site, this specific area, so we put it here with specific with ultrasound, basically using ultrasound to increase this site, okay, this site, this area, to increase its temperature. So maybe it's more than 40 degrees Celsius. So the changes of temperature will specifically in that particular area, right? Because due to the ultrasound, you have megahertz will increase your temperature without damaging your skin tissue and so on. So that's why we use ultrasound. So once the liposome arrive, okay, towards or lalui lah, kita kata lalui, without arrive tu dia akan melalui that specific area. So the changes of the liposome in terms of due to it come to the more high temperature, so it will be triggered. Once it will be triggered, okay, so it will be the drug will be released that particular site. Is it okay? Am I answering your question?
Yes, doctor.
Okay. All right. So usually the thermosensitivity, because the heat of that specific is not damaging, it's been opened up. Okay, we know that temperature will increase its reaction. Okay, will we increase the stability. Okay, we know that the example other than liposome is, we know that if we want to dilute our sugar in a high temperature is more easy compared to lower temperature. So that's why the idea of increasing the temperature will be increasing its reaction towards that particular temperature without damaging the drug itself. So okay, that's the idea of thermosensitive liposome. Okay, is it okay?
Okay, doctor.
All right.
Thank you, doctor.
Okay. So we continue, I think. The last part, I think the last part of the liposome drug delivery system is targeted delivery. Okay. Okay. So targeted delivery liposome should selectively localize anti-cancer drug at the tumor site, thus reducing the toxicity of the drugs to normal cells and improving their therapeutic activity. So meaning it will deliver to the therapeutic site without it being delivered to the healthy site or healthy cells that can be at the end can some kind of damaging that particular cells. Okay, that's why we have targeted delivery system. Various antibodies have been conjugated to the surface of stem liposome, within okay, this is related towards the stabilized liposome, the first one, to produce so-called immunoliposome for active targeting. And studies have shown that accumulation of immunoliposome at the tumor site does not necessarily increase the therapeutic activity compared to liposome without antibodies. Okay. The main reason is that the drugs cannot escape from the carriers after reaching the tumor site. Tumor-specific targeting is critical goal in research on liposomal drug delivery. So meaning we deliver the site, and the idea we don't want that particular drug to be delivered other than our tumor site, right? The tumor area, because of because we want to ensure that the minimum concentration, we want to ensure there is no because there is no interaction with it, other system, other healthy cells. So that's why. So monoclonal antibodies against tumor-associated antigens have been successfully adopted for targeting cancer cells. And tumor-specific ligands other than monoclonal antibodies have also been investigated as in vivo tumor-directing molecules. Okay. Besides antibodies, ligands for cell surface receptor are also used as active targeting groups. Okay, tumor cells contain increased numbers of transferrin receptors compared to normal cells, and transferrin has been used as a ligand for delivering anti-cancer drug or drug containing liposome. I think you are well aware about the ligand. Ligand is term house gas molecules, meaning that particular ligand will trap some kind of gas molecule. So the ligand will be the transferrin. So the drug will be the gas. So they will attach together usually without covalent bond. So this called a ligand reaction. Okay, I think you are aware this is uh, inorganic chemistry, right? Other ligands such as carbohydrates have been used to target selectin and other cell surface receptor. So specific targeting to internalizing cell surface receptor has increased both in vitro cytotoxicity and in vivo efficacy of liposomal drugs. Okay. All right. That's all. Thank you, class, for this topic. Any question?
Doktor,
Yep.
saya ada satu question lah. Cuma untuk kan macam liposome kan dia consist of phospholipid. So kalau if let's say kalau satu drug tu sebab kalau drug tu yang macam dia kuat di semipolar doktor. So dia akan duduk dekat tengah-tengah region ke ataupun dia boleh campur juga dekat mana-mana dia boleh scattered dekat hydrophilic dengan hydrophobic kalau di semipolar.
Okay. It can be, okay, the idea awak tengok yang the sphere, okay, sphere here, right? Jap. I think if I show here, you can easily visualize the idea of it. Okay, right? Okay. So meaning they have both. They also as the drug also is amphiphilic, is it? And
Yes, doktor.
right. So the both also amphiphilic. So in terms of without the drugs, you imagine without the drugs here, without the drug itself, it will also form a sphere. Okay, without the drug. So meaning the center will be empty. Okay, so still the inner side of here will be hydrophobic. So again, I mentioned before, the idea is based on light dissolve light and something like that. So the hydrophobic will attach to hydrophobic segment. So because of the amphiphilic drugs that mentioned by they've been asked by Amin. So again, due to hydrophobic entrapment, so again it will be entrapped here. Even though it has amphiphilic segment, but due to hydrophobic interaction. So the inside the sphere here, the hydrophilic will not be tak berguna sangat lah, because the interaction is due to hydrophobic interaction. So how about the outside treatment, outside sphere? So there's a possibility that they will attach towards the drugs, okay, towards the drug. However, is it the number encapsulation is quite high? No, because we know that the interaction is non-covalent interaction, so it's quite weak basically. Okay, quite weak basically, so it can easily be triggered or can be loosened. Okay, so due to the surface, okay, we can say that this sphere, the outer sphere, is only the interface between the outer segments of the whole system. So basically it can easily be released. Okay, it can be easily be released. Okay, but in the end we still can entrap quite number amount of that particular drug within the system. Do you understand? I mean, am I answering your question?
Yes, betul.
Kan?
Faham betul.
Boleh visualize kan? Right?
Boleh visualize, boleh.
Okay, it's not been scattered. It can be scattered, it's possible that particular drug can be attached here, but still at the end it can be entrapped due to hydrophobic atopic interaction. Okay?
Alright. Thank you, Doktor.
Okay. Right, any question? Any more question?
No, Doktor.
Tak ada. So far so good?
So far so good, Doktor. No question, Doktor.
Nervous?
No questions.
Oh, no question. I thought nervous. Nervous ada record kan?
Tak
Alright,
ada.
I think before that I stop recording before we go to borak-borak ke apa kan? Kena
Ni saya kat bilik, Doktor. Tak ada dalam bahal kat bilik ni.
Okay. Tak
Doktor
apa, men.
risau.
We stop recording first. Okay.